Hello everyone, how you doing? Welcome again to Total Immunology. And in this episode, I'm going to try and continue something that we started in previous episode, where we asked the question about what is a healthcare laboratory. In this episode, we're going to start to look at what does an immunology laboratory do. Okay, so we're going to leap straight back in now and look at some slides that help you show this thing that we're talking about. So when we talk about healthcare laboratory science, taking it forward from where we took last time, we're going to talk first of all about one group called the blood sciences. It's a bit of a misleading name, if I'm truthfully honest, because it's not the only type of sample or the type of substance that these laboratories will measure, but we call them blood sciences because that's predominantly what they measure. Okay. And inside blood sciences, there are three disciplines that you will probably know and probably recognize. Okay. So, number one of those is biochemistry. It has the biggest workload and it does has the biggest array of tests and the biggest demands on it. And really, what biochemistry does is exactly what I've written there. Okay, it measures different ions and substances in the body, which are both intrinsic and extrinsic. What do I mean by intrinsic and extrinsic? Intrinsic means they are things that are natural or internal to you. So they are produced by you, they are something that you make. So, an example could be a hormone. Extrinsic means it is something that's not part of you. It's something from the outside world that you may, in the case of biochemistry, have put inside yourself. And that could be a drug, both medicinal or recreational. The next one we have is hematology. Hematology is a bit more confined. And what hematology does is measure and characterizes the components of the blood system. So that could be the cells, it could be the substances that help make those cells, like hemoglobin, for instance. It could be looking in different parts of the body, both the blood or in something like the bone marrow, where a lot of the cells, particularly the white blood cells as well, all come from. And the final part, which is the bit that's dearer to me out of the blood science groups, is immunology. And immunology, again, is a little bit more defined like hematology in the sense that it measures and characterizes the components of the immune system. So these aren't the only labs that we have in healthcare laboratory science. We next have after that genetics. And what genetics' job is to do, very simple, and it's clues in their name really. They characterize part of the genetic sequence of an individual, and that can either be germline. What do I mean by germline? I mean that it was a piece of DNA that they were born with, a genetic sequence that was made with them the moment they were born, or it could be somatic. And what does somatic mean? It means the opposite to that. It means that a somatic gene sequence is something that has been developed over time. And so the best example of that is going to be cancer. If we have a situation where someone has a tumor in them, we may want to take out and genetically sequence that tumor or the cells of that tumor to be able to understand what DNA that tumor has that could indicate why it is a cancer. Why is it cells from the person that have become cancerous? Okay, so that is genetics that have something different but complementary to what we do in the blood sciences. We have two more areas to look at. The next one is cellular pathology. This is one of the oldest parts of healthcare laboratory science. And what's their job? Their job is to measure and interpret the appearance and composition of different pieces of actual tissue. So they're dealing with the largest kind of macro structured part of the human body, because what we have to do really, and we want to get a bit medieval about it, is we're going to go in and we're going to cut something out and remove something from the person. In the sense, we're going to take a piece of an organ or the piece of this of tissue or structure of the person, and we're going to now look at that in detail, usually on a slide. Usually, we're going to embed it in wax, or we're going to stain it so that we can see the contrast. And we're trying to look at that to understand what it looks like and why might that be linked to the symptoms that the person has. The final group that we have is microbiology. And so they are slightly different to all the others here, because in all the other cases here, we've been focusing on the patient and we've been looking at what is it about the patient or what the patient has taken or interacted with in the outside world that we can measure. Microbiology, it's exclusively about the idea of us trying to take samples from a patient to understand if there's evidence that the patient has a microorganism of some form in them, on top of them, around them, or if they have evidence that they've had prior exposure to that microorganism. So when we talk about viral serology, what we're often doing, if you ever hear that term, is that means that we're trying to look for evidence that the person has seen a virus in their past, and that could be linked with vaccine responses. So at the moment, COVID is very big at the moment. We have lots of COVID vaccines flying around. One part of microbiology's workload is to look at for evidence of COVID serology, to see evidence that the body has successfully seen COVID. Okay. Now there are overlaps across all of these different disciplines. And so there may be one center in the world that says that they do a lot of genetics, even though they're an immunology lab, or they do a lot of hematology work, even though they're a genetics lab. And that's because we have lots of crossover disciplines all over the place here. So I don't want anyone to think these are firm boxes. These are appropriate regions of how we divide up the work and what the point of each of these sections is. So let's now look at immunology particularly here and say, what sample types do we measure? So the biggest sample type we measure is blood. I've given you a sample of a blood tube there. There are lots of different types of blood tube. So although the blood that comes out of the patient is the same, actually we do different things once the blood enters the tube. Sometimes we let the blood just clot, which is the most natural thing it can do, and we take from that blood the serum, okay? Or we might put in it into another tube where it doesn't let the blood clot, because we want the blood to be whole blood. We want the cells and the proteins, the serum proteins, to be able to be all mixed up together so that we can measure certain things. The other type of sample type we might have is urine. Okay, much less common than blood, but we still do measure urine. And again, we're trying to look for evidence of particular components of the immune system inside that urine. And the final one that we look like again is sort of CSF, stands for cerebrospinal fluid. And so cerebral spinal fluid is a type of fluid that travels through and is where you store a lot of proteins that run through the central part of your nervous system. So all the way along up your spine and into your brain. And that is the fluid that is used to communicate and give nutrients to that part of your body. To be able to get hold of that sample is the hardest one out of all three of these. Urine's the easiest because we just go for a wee. Blood is the next easiest because we can relatively easily catheterize a person to get blood out. And CSF is the most difficult because we have to do something called a lumbar puncture to be able to isolate that out. So in what way does laboratory immunology measure? Okay, we have all these samples, we can do different tests, and what types of results can we generate? So the first type of result we can generate is something called qualitative. What does qualitative mean? It means very simply that we're giving either one or the other answer in the majority of the time. So we might say something is positive or something is negative. We might say that it is correct or not correct. Okay, so we might say that it's a yes or a no. Okay, we might say that it's normal or abnormal. In each of these situations, you can see that they're all really one and the same thing. We're saying it's either good or it's bad. That's really all we're saying. And that's all we can say at that point with that type of test. We can't interpret it any more than that. We can just say those things. But the other type of test we can do is something that will produce a result that's quantitative. So when we have something that's quantitative, it's pretty obvious what that means. It means we're going to generate some sort of number. Now, generating a number, and I speak as from personal experience, can be something that both educates and stresses out people all at the same time, because a number is only as good as the context or perspective you have to put it against. What do I mean by that? If I show you here the situation where we now put some of these numbers that we have on the screen, and we now start to put them into context about what does that look like, you can see here I'm saying, if your sample on this test produced a value that was between 9 and 25 on our test, then that means that you're going to have an answer that is the same amount as what most healthy people would expect to have of whatever it is that we're measuring here in our example. If you happen to have a value that comes out below 9, then we're going to call that an abnormally low amount of it. And obviously that can mean the other way around. We said it's above 25, then we're going to say it's abnormally high amount. So what we have here, in order for a quantitative result to truly work, we actually need to have a reference range. And a reference range is understanding what is difference between normal and abnormal. And exactly how we do that, I'm going to have to put in a separate talk in the future, I imagine, or I may have already done it, depending on when you watch this. Look in the corner and you might see a little link. But what we need to always understand about this is, first of all, what is the reference range? And secondly, what is the gap between our result and the reference range? And what is the total measurable range we could even measure on the assay? Because if we can measure up to 10,000 and you've only got three numbers above the top end of the reference range. So the reference range is up to 25, like this example here, but you've now got a value of 28, but we can measure all the way up to 10,000. Does 28 really mean anything that serious? Because it's only just above, okay? So we have to always think about a number in the context of the numbers that we know from what we've done in experience of normal people and abnormal people, and people who may have very clear-cut diseases as well. So now we know what kinds of way we can develop results, what samples we can use, what type of labs we have in healthcare science. What actually does immunology measure, and how can we divide that up? So I've got for you here a schematic of how we can divide up the different types of measurement that we do in laboratory immunology according to what we want to try and understand. And then we'll move on from there. So the first and probably the biggest part of our repertoire of tests that we run in an immunology laboratory is to do with autoimmunity. And what is autoimmunity? Basically, what we're doing, if we're testing for autoimmunity, we're trying to measure for evidence that the immune system is creating something that is directed against the body itself. And that the very way that it's done that has led to now the immune system causing damage to the patient. So that's really what autoimmunity is. And when we do an autoimmunity test of some form, and there are many of them, that's really what we're trying to do. And depending on the test, that evidence that we find will be either very, very strong to indicate that the person has a particular autoimmune disease, or it may just be one single piece of evidence and we need multiple to be able to make that decision. The next big area that we do is immunodeficiency. What does immunodeficiency mean? Immunodeficiency means that we're going to try and measure different immune components, okay? So different parts of the immune system that we know about, and we're going to try and see if they're completely absent. We're going to try and see if they're low, or we're going to try and see if they are there, but they're non-functional. So three different categories we can put these things into. We can also try and understand whether a person is primary immunodeficient, which means that they were born with a particular difference to them, usually genetic, that means that they can't make a particular part of their immune system or it doesn't work. Or it could be secondary immunodeficiency, which is where they may have something else going on in their lives, which inadvertently is leading to their immune system now having one of these problems. And so there are many different causes for secondary immunodeficiency. There could be drugs, it could be infection, it could be cancer, for instance. It could be other things about their body, like their kidneys are damaged. We will talk about that again in another separate episode. The next thing we have and we measure is allergy. Now, allergy is a quite a contentious word in the world of immunology, so I'm going to say it very clearly what I mean about the word allergy here. Allergy in this case is where we're going to measure for evidence that specific parts, not just any part of the immune system, but very specific parts that we associate with classic allergy, are creating an immune response against specific harmless extrinsic targets. Okay, so they're targets that are outside of the body, they're not part of the body itself, they come from the outside world, and we would normally consider them to be harmless. They're things that are not going to cause problems to the normal person. So, an example might be peanuts, another example might be tree pollen. These things, if you are allergic, classically allergic, could lead to you having an allergic response. And there are many different types. And again, we will look at this as we go through the channel and develop the channel. The next area we have is thinking about immune system dysregulation. This is probably the hardest one for me to try and define, again, without treading on the toes of established immunological words. So this, again, I guess is quite controversial the way I'm describing this, but hear me out. If you're someone who knows immunology and want to challenge this, then fair enough. But what we're trying to do with dysregulation is we're trying to measure for evidence that the immune system is over-responding to extrinsic targets. Okay, so extrinsic targets, again, these outside elements, but in this particular situation, those elements could be harmful. They could be something like a pathogen. Okay, so it could be a fungus, for instance, that normally is something that will try and infect a human being and live inside a human being, but most people are able to cope with that and not let that fungus attack them or cause them problems. But some people, they don't get the infection, but instead what they get is an incredibly over-responsive immune system. Now, uh, there's a word that I'm not going to use in these talks, which is hypersensitivity for this, because I'm saving that word to define lots of different ways that we look at immunology in a later episode. But you could say these people are overly sensitive or hypersensitive, I guess. The final part that we look at is malignancy. We do not look at all types of malignancy by any stretch of the imagination, but we do measure in immunology commonly. We measure for evidence of components of the immune system that have become cancerous. And in particular, we're looking at B cells in immunology, and we're normally looking at a particular type of B cell called a plasma cell. Okay, and again, there will hopefully be links, we'll link into where this goes in the future. But that's just to bring out another area that we'll often see in an immunology laboratory lab. Now, that doesn't mean we only do 1, 2, 3, 4, 5 tests by any stretch of the imagination. If I take the immunology service that I'm involved in, we have around 70 different tests that we have in our catalog that we can measure for people. And in those 70, there are in one particular case, you can see here in allergy, we have this one test called specific IgE. Just to put it into context again, the specific IgE test, there are around 250 variants of the specific IgE test. So it isn't a case of that just being one single thing that we measure. We can measure it in many, many, many different ways, but that is the name of that test. And it's the most important part of laboratory allergy testing. You can see all these other different tests that we do in these different areas. You can see in the autoimmune area, we do a lot of things that are called screens. And again, in each of those screens, we will commonly have multiple separate tests that have been developed over from different parts of history and origin that have come together as we understand the conditions that they're surrounding to be able to form a particular investigation screen that we would use. And in immunodeficiency, what we're doing there is measuring for those particular components of the immune system, and we're trying to, as I said, quantify them, maybe test their function. Okay, so hopefully that's been useful for you. So what we have here is a nice description of what an immunology lab does, putting it into context of other healthcare laboratories, and understanding the beginnings of how we look at samples, what we do with them, and what kind of results we generate on them. I hope that's been useful. I wanted this to be quick, and I hope it has been. I'll check my watch in a minute. Keep yourself well. Please subscribe, like, and comment, and tune in for future episodes as they come about. Thank you very much.